coming back to our classes. Now, in the last class, alcoholic liver disease was the major cause of cirrhosis. So let us see what this alcoholic liver disease is all about. So ethyl alcohol, it is the most common cause of acute and chronic liver diseases, and almost 70% cases of cirrhosis are because of alcohol. Alcoholic liver disease, we can study this disease in three overlapping syndromes. The first one is your hepatocellular steatosis or fatty change, or it could be the second one is your alcoholic steatohepatitis, and the third one is steatofibrosis, which ultimately leads to cirrhosis. Now, when you talk about steatosis, so steatosis is nothing but cell degeneration in which there is excess of accumulation of the fat in your hepatocyte. Because we are calling it as hepatocellular steatosis, it means there is accumulation of the fat in the hepatocyte. And the second one is that this accumulation is leading to inflammation of the liver, which is called as alcoholic steatohepatitis, and this ultimately is leading to fibrosis, which is steatofibrosis, and when it is extensive, diffuse, and it is spanning the whole of the liver, we call it as cirrhosis. Now all the stages are reversible except cirrhosis. Even the steatofibrosis stage also, the scars can reduce, right? So this is what is alcoholic liver disease. Before going further into alcoholic liver disease, let us understand the metabolism of ethyl alcohol. Ethyl alcohol or the ethanol in the hepatocyte cytosol. What is cytosol? It is nothing but your cytoplasm, right? Here, in most of the ethanol, almost 60 to 70% of your ethanol, alcohol dehydrogenase converting the NAD into NADH causes the formation of acetaldehyde, and another 70% is by this alcohol dehydrogenase. A little of it is converted into acetaldehyde in the microsomes. Here NADPH is converted in NADPH is reduced in these cases. And finally, lastly, the peroxisomes, which are present in the hepatocytes in the presence of catalase, will cause the conversion of your ethanol into acetaldehyde in the presence of hydrogen peroxide, but this occurs only in 5% of these cases. This acetaldehyde is further converted into acetic acid in mitochondria in the presence of alcohol dehydrogenase. Aldehyde dehydrogenase. This is aldehyde dehydrogenase. So the ethanol gets converted to acetaldehyde and then next into acetic acid with the help of the enzymes 60 to 70% by alcohol dehydrogenase in the microsomes by the microsomal enzymes, and in the peroxisome only 5% of the alcohol is converted into acetaldehyde using catalase, and this acetaldehyde in the mitochondria is further converted into acetic acid. So the acetaldehyde, which is the intermediate metabolite, is the hepatotoxin which is present. And not only this, acetaldehyde is a hepatotoxin by itself. Not only this, there is a diversion of the metabolism to the alcohol. So there is a diversion of all the metabolisms towards lipid accumulation. Now, The fatty acids which enter from your splanchnic circulation are the oxidized and acetate is being formed. Then you have phospholipid formation, cholesterol esterase formation, triglycerides and lipoprotein formation, and they get secreted. But when you are having an excess of alcohol intake, all this lipid gets accumulated in the liver cell. So from the diet, from the peripheral lipolysis and de novo lipogenesis, there is excess of fatty acid formation. This fatty acid is oxidized to acetates. Some of them is oxidized in the peroxisomes, and the triglycerides are converted to VLDL. So coming to alcoholic liver injury, the acetaldehyde is the hepatotoxin, and the diversion of the fat metabolism to alcohol leads to fat storage. There is an increased peripheral release of the fatty acids, and the fat storage and the fatty change. This is what is occurring in your liver. When there is an excess of fat storage and the swelling, there will be rupture, which leads to fat necrosis and severe inflammation, and inflammation is always followed by fibrosis. And when this is occurring again and again and again, and the whole of your organ gets fibrosed, it is called as cirrhosis. So alcohol stimulates collagen synthesis, inflammation, portal bridging, fibrosis and micronodular cirrhosis. This is what is in short alcoholic liver injury, pathogenesis of the alcoholic liver injury. Now coming to the first entity, the hepatocellular steatosis, or the fatty change of the liver. It is the most frequent and the earliest thing, and it is because of the direct toxic effect. So what is the toxin here? It is the acetaldehyde. There is an increase in the triglycerides, increase in the fatty acids, decrease in the oxidation of the fatty acid, and decrease in the formation of the lipoproteins. So when there is decrease in secretion of these fatty acids, what is happening? It gets accumulated in your hepatocyte, and this is leading to the fatty change in your hepatocyte. Initially, these fat globules are stored as small vesicles within the hepatocyte, and this is called as microvesicular fatty change. As the amount of the fatty acids get accumulated in the cytoplasm, bigger vacuole is formed, and this is called as macrovesicular fatty change. And when there is an excess of accumulation and the surface tension of your hepatocyte cell membrane is getting more, there is a rupture of this, which leads to a granuloma formation, and this rupture also causes inflammation and fibrosis, and it is perivenular and perisinusoidal fibrosis which it starts as. Yes, steatosis. When there is accumulation of all the fatty acids, it becomes a steatotic cell, hepatocyte, hepatosteatosis. And this accumulation in the liver or at the gross level, it shows that there is an increase in the size of the liver, and there is this yellowish glistening appearance of the liver grossly. Yeah, the yellowish hue which is there, and the weight of the liver and the size of the liver, or there is an enlargement of liver. Now these are your fatty change. So the hepatocytes are actually being transformed into fat like cells. This is what is called as the steatotic change. And look at this, there are two cells which coalesced to form a fatty cyst, and this, as it increases in size, it ruptures and forms the lipogranuloma. These are all your fatty change. So it can resolve completely if alcohol is stopped. Now the fatty change what happens in your alcoholic hepatitis, which is steatohepatitis. That is the second phase. Alcoholic steatosis is only accumulation of the fat in your hepatocytes, whereas alcoholic hepatitis, there is a step further where this fat is leading to inflammation of your liver. First change is your fatty change, just like how we saw it in the steatosis. Then there is a further second thing. What happens here is ballooning degeneration of the cells. The third change what we find in alcoholic steatohepatitis is you are having focal necrosis. Necrosis is always followed by inflammatory cell infiltration or the inflammation. And here the intermediate filaments get accumulated, and these are called as alcoholic hyaline. There is fibrosis in the perivenular or the pericellular phases. And why does this fibrosis or the inflammation occur? One is the immunological attack. The second one is because of the free radicals which are causing if you remember, alcohol is causing an excess of it is causing an excess of bypassing of methionine formation, which is a major thing which is required in the glutathione, which is actually a free radical injury scavenger. And when that is lost, there is membrane proteins which are getting attacked by these free radicals, and this causes hepatitis. And acetaldehyde by itself, it causes lipid peroxidation and adducts formation. What is adduct formation? That is, there is the proteins which are present are getting instead of being dispersed, they get together and form an adduct formation. So this is your fatty liver, alcoholic fatty liver. Alcoholic steatohepatitis, it is characterized first by hepatocyte swelling and necrosis, and this is called as ballooning degeneration and necrosis, and this is because of the accumulation of the fat as well as water. This ballooning degeneration is because of accumulation of fat and water. That is the difference between your ballooning degeneration and your steatosis, which has occurred in alcoholic steatosis. There is only fatty change, that is steatosis. Here also the water is getting accumulated, and that is causing your ballooning degeneration. Yes, this is the degenerated cell. Yes, all these where your cytoplasm gets ruptured, and the structure, if you look at your normal cell, you have a variable staining of your cytoplasm. In a necrotic cell or the lysed cell, all that is broken up. This is what is lysis. And then the second characteristic point in alcoholic steatohepatitis is that there is formation of Mallory-Denk bodies. What is this Mallory-Denk bodies? These are amorphous eosinophilic material present in the hepatocytes, and this is because the intermediate filaments, they get complexed with ubiquitin protein, which is ubiquitin is a protein which helps in removal of your of your senescent molecules. This ubiquitin protein, it complexes with these keratins and they get agglutinated, and they form the Mallory-Denk bodies. Here intermediate filaments are the cytoskeleton. Just like how we have the skeletal system to maintain the structure of the body, the same way we are having the cytoskeleton which maintains the structure of a cell. These intermediate filaments have to be spread out and hold the cell in place. But when you are having an excess of alcohol, when alcoholic injury on the cells, these intermediate filaments, they get agglutinated and they complex with this ubiquitin protein, and they form these Mallory-Denk bodies. And this Mallory-Denk bodies, they are usually seen in alcoholic steatohepatitis, but it is not specific for this because they are seen in other liver diseases like the Wilson's disease. See, this is your Mallory-Denk body. Just like how we have a skeleton to hold our body and the muscles in place, similarly your tissue is having the extracellular matrix and extracellular matrix actually, which is holding the cells apart in the way they have to be present. In the same way, your cytoskeleton is are filaments which hold different structures of the cells, like your ribosomes, your Golgi apparatus in place. Those intermediate cells, because of the alcohol or the acetaldehyde injury and the free radicular injury, they get agglutinated and complex with the ubiquitin protein. This is what is the Mallory-Denk body. And then we also, the third one is neutrophil infiltration. See, look at this. These are the neutrophils. This is the nucleus of a hepatocyte. This is the nucleus of a hepatocyte, and these are the neutrophils. What are neutrophils? Which have segmented nucleus. So this is a new neutrophil, this is a neutrophil, this is another one, this is a neutrophil. So there is neutrophil infiltration into the hepatic lobule, and they accumulate around the degenerating hepatocytes. So what are the three features of your alcoholic steatohepatitis? The first one is necrosis, which is the ballooning degeneration. Alcoholic steatosis, that is the fatty change is present in alcoholic steatosis. What are the additional changes which you find in hepatitis? The first one is your ballooning degeneration, and this degeneration is because of not only accumulation of the fat, accumulation of the water as well. Then there is necrosis, then we have the Mallory-Denk body formation, and the third one is the neutrophilic reaction, neutrophilia. This is the alcoholic hyaline formation or the Mallory-Denk bodies. This is to show you the infiltration by neutrophils. This is the steatosis, followed by degeneration, and followed by your neutrophilic infiltration of the tissue. This is what is alcoholic hepatitis. Alcoholic hepatitis or alcoholic steatohepatitis is, in addition to the fatty change which occurs in the liver, you also have ballooning degeneration, which is accumulation of the water leading to degeneration of the cell, and necrosis, ballooning degeneration and necrosis of the cell. The second one is the intermediate cytoskeletal filaments. The intermediate filaments, which are the part of the cytoskeleton, they are getting agglutinated, just like that is, they are coming together and forming a clump. They are the Mallory-Denk bodies. The third one is the neutrophilia into the tissue. And the third one is called as alcoholic steatofibrosis. Look at this. This blue color is called the fibrosis, and this is the central vein. Around the central vein and in between the cells, pericellular centrilobular fibrosis is what is alcoholic steatofibrosis. Why is it occurring only in the centrilobular region? Because this is the area where we are having the maximum of alcohol detoxification which is occurring. So alcoholic steatofibrosis begins in the centrilobular pericellular regions. So prominent activation of sinusoidal stellate cells. Where are they? These stellate cells are nothing but the Eto cells which are present, which we have seen in the previous class, which leads to the formation of which leads to the transformation of these Eto cells into the myofibroblasts. And fibrosis begins with the sclerosis of the central vein. What is sclerosis? It is nothing but change of, there is an excess of extracellular matrix being produced, and deposited around your veins. That is the sclerosis of the central vein. And then there is perisinusoidal scar that accumulates in the space of deceive. Space of deceive is in between your hepatocyte and your endothelial cell of the sinusoid. The space of the deceive of the centrilobular region, spreading outward and encircling the individual or the small clusters of the hepatocytes in the chicken wire fence. So the blue lines which are present around the central vein and extending in the space of the deceive in between the hepatocytes or in encircling groups of hepatocytes. That is what is the steatofibrosis. And so acute liver cell necrosis after a bout of heavy alcohol drinking superimposed on steatosis and fibrosis after 2 to 3 years, it leads to cirrhosis. So this cirrhosis is termed as Laennec cirrhosis, and perisinusoidal scar eventually link, as it keeps on increasing, it links the portal tract by fibrosis, and then there is a central to portal fibrous septa formation. It is the fibrous septa which is formed in the cirrhosis. With developing nodularity, cirrhosis becomes established. From where do this nodularity appear? It is from the liver stem cells. This is the these are the micronodules which are present in your alcoholic cirrhosis, which is also called as Laennec cirrhosis. Yes, these are the regenerating nodules, and the blue staining is all the scar tissue formation. So what is alcoholic hepatitis? The clinical features, most of them are asymptomatic and they usually complain of vague abdominal discomfort, and jaundice, fever, nausea and neutrophilia occur in rare cases. Only 10% of them end up in death, and 1/3 of these cases end up in cirrhosis. So fat rupture, fat necrosis, severe inflammation and fibrosis leads to alcohol stimulation collagen synthesis, inflammation and portal bridging fibrosis, which leads to micronodular cirrhosis. Now coming to alcoholic cirrhosis, it is to 60 to 70% of all the cirrhotics are because of alcohol. Yeah, among the cirrhotics, if you see, 70% of them are because of alcohol, but only 15% of the alcoholics become cirrhotic. This is to show you a fatty change, which is enlargement and yellowish glistening appearance of your liver. This is, along with that yellowish discoloration, you also have nodularity, micronodules. This is the normal liver, and this is to show you the glistening appearance of the liver, the yellowish discoloration and the micronodules. This is cirrhosis with fatty change. Yes, micronodular cirrhosis, extensive micronodular cirrhosis. These are the scars or the fibrovascular septae. These are the nodules, and within the nodules you are having the steatotic or the fatty change. Alcoholic cirrhosis, again most of the cases are asymptomatic, and the other features of the cirrhosis come in, like hepatic coma, massive hemorrhage of the esophageal varices. You can also have infections. We have hepatorenal syndromes and hepatocellular carcinoma developing in the cases of the cirrhosis. And coming to the laboratory findings, usually there is an increase in the aminotransferases, increase in the bilirubins. Initially, first is your aminotransferases which increase, then bilirubin, alkaline phosphatases are increased. You have anemia and hypoproteinemia later in the course of the disease. So the normal liver, first stage is steatosis, and in the cases of abstinence it can revert back to normal, or it can go to steatohepatitis, which is, these are all interchangeable actually, the steatosis, steatohepatitis and cirrhosis. It is not necessary that all the times the patient has to go to the stage of steatosis and then have steatohepatitis. Initially itself it can cause steatohepatitis without this intervening stage of steatosis. So prognosis in the alcoholic cirrhosis, in the 5-year survival rate, if there is no jaundice, if there is no jaundice, no ascites, no hematemesis, and they abstain from alcohol, it is almost 90%, 89% of 5-year survival, but it reduces if they keep on taking alcohol to 62%. If you are having jaundice, it is less. When you are having jaundice, it is only 57.5%, and ascites also it comes to close to 50%, and when you have hematemesis, it reduces to 35%, and these rates come down much more when you are having alcohol. So jaundice, ascites and hematemesis are taken into the consideration for prognosticating cases of alcoholic cirrhosis. So alcohol leads to fatty change of the hepatocyte, and this Mallory-Denk bodies which are getting deposited, and these Mallory-Denk bodies are nothing but accumulation or conglomeration of your intermediate cytoskeletal filaments, and then when you are having a necrosis of this fatty change with neutrophilia, it is alcoholic hepatitis, and repeated bouts of inflammation leads to fibroblast, the Ito cells being converted into myofibroblast, which leads to the initial phase is called as steatofibrosis, and finally extensive fibrosis leading to cirrhosis. So alcoholic steatosis, alcoholic hepatitis, and cirrhosis of the liver. We also have fatty change not only secondary to alcohol, it can also be secondary to diabetes, obesity, or severe gastrointestinal malabsorption, and this is termed as non-alcoholic steatohepatitis. This is a little older term. Nowadays we call it as non-alcoholic fatty liver disease. So what is the mechanism of hepatosteatosis? In alcohol, the acetaldehyde is a hepatotoxin which alters the mitochondrial and the microsomal function. The other causes like carbon tetrachloride, it causes protein malnutrition, decreasing the synthesis of apoprotein from the peripheral stores, which lead to hepatosteatosis. Any doubts or any clarification, please reach me on this mail. Thank you.